Welcome to the deep dive. You know, right now, if you receive a life-saving organ transplant, your own body well, it essentially treats that new tissue like an invading army.
Right, it mounts this incredibly hostile, coordinated attack.
Yeah. And that war, the organ rejection, is still the single biggest hurdle in clinical transplantation today. But imagine if the donor organ could deploy this invisible fleet of microscopic stealth ships.
Like a covert operation inside the body.
Exactly. Stealth ships designed to just dismantle that immune response before the first shot is even fired. Today, we are pulling from a pretty dense stack of sources. We're talking cutting-edge immunology research, medical journals, transplant studies.
There's some genuinely paradigm-shifting stuff happening in intercellular communication right now.
Oh, absolutely. So our mission today for you listening is to explore these things called exosomes. Specifically, we're looking at how the deliberate absence of these ID badges proteins called MHC class 1 and class 2 on purified exosomes minimizes the risk of host-graft reaction.
Which basically allows these tiny vesicles to fly completely under your immune system's radar.
Right, because traditionally, to stop rejection, doctors just glottally suppress your entire immune system, right?
They do, yeah. Which leaves the patient incredibly vulnerable. But this research, it looks at nanoscale interventions. It's about manipulating the body's own communication networks to create localized tolerance.
So it's not a carpet bomb, it's like a sniper approach.
Exactly.
Okay, so to understand these invisible delivery trucks, we first need to get a baseline of how cells talk to each other and, how that communication accidentally triggers these massive immune wars.
Right, and that brings us to exosomes. Our sources focus heavily on these. They are extremely small extracellular vessels. I mean, we're talking 30 to 100 nanometers in diameter.
Which is unimaginably tiny.
Very. And almost all cells in your body secrete them. They carry this highly complex cargo proteins, lipids, messenger RNA from a parent cell to a recipient cell.
So they're microscopic mail trucks delivering biological packages.
That is a perfect way to think of it. And their origin story is actually fascinating. They don't just pinch off the surface of the cell. They form deep inside within these things called multivesicular bodies.
Okay, so they're built internally before they're ever released.
Yes. They fuse with the outer membrane and then get released into the extracellular space. And that specific endosomal pathway is why they're so potent.
Because they take pieces of the parent cell with them.
Exactly. The membrane of the exosome incorporates the exact surface proteins of its parent cell. And in allotransplantation, getting an organ from someone else, the most critical surface proteins carried over are the MHC antigens.
The major histocompatibility complex.
Right. Your immune system uses these highly specific proteins to tell the difference between your native healthy tissue and something completely foreign.
Okay, let's unpack this with the mail truck analogy. If exosomes are the delivery trucks, then the MHC proteins are basically the license plates or like the driver's ID badges.
That's spot on.
And if the immune system's security guards, the T cells, spot a truck with foreign license plates, they just sound the alarm.
They recognize the alanogen, bind to it, and initiate a total rejection cascade. Yeah. It seems super straightforward, right? Recognition and attack.
Right. Bad guy spotted, bad guy attacked.
But the literature on how your immune system actually intercepts these foreign markers has evolved a lot. For decades, we relied on the passenger leukocyte concept.
Which is the direct pathway, right?
Yeah. The idea was that donor white blood cells, physically trapped inside the transplanted organ, migrate out, travel to the recipient's lymph nodes, and just directly show their foreign MHC to your T cells.
Just walking right into the police station.
Basically. But those donor cells don't live very long. When they die off, the indirect pathway takes over, where your own immune cells scavenge the debris and present the fragmented foreign proteins on their own native MHC.
Okay, so they hold up pieces of the dead invaders to warn everyone.
Right. But the sources highlight a third mechanism, and this completely rewires how we understand rejection. It's called the semi-direct pathway.
And this is where it gets really interesting, because this relies on those exosomes.
It does, and it's driven by this bizarre phenomenon scientists call allo-MHC cross-dressing.
Cross-dressing, like biologically.
Literally, yes. In this semi-direct pathway, the healthy cells of the new organ continuously secrete their exosomes, which, remember, are covered in the donor's foreign MHC.
The foreign license plates.
Exactly. They travel through the bloodstream until they are captured by your own antigen-presenting cells, your APCs.
And normally your APCs would just eat them and destroy them in a lysosome, right?
Normally, yes. But here, instead of degrading them, the recipient cell undergoes a membrane fusion event. It takes the intact foreign MHC molecules straight from the exosome and embeds them into its own surface.
Wait, right alongside its native proteins?
Right alongside them, your own dendritic cells and macrophages are actively integrating the foreign identifiers.
Wow. So let me get this straight. You're saying the listener's own native immune cells are basically putting on the enemy's uniform, parading around the lymph nodes, and subsequently getting attacked by their own immune system.
That is exactly what happens. Biologically, this evolved so immune cells could share antigens and fight viruses better. But in a transplant, it causes a fatal biological misunderstanding.
Because the T cells don't know it's a friendly cell in a stolen jacket.
Right. T cells lack the capacity to distinguish that. They just react to the foreign structure and launch a catastrophic immune response against your own cross-dressed cells.
That is wild. The transplanted organ is inadvertently weaponizing our own communication network against us.
Exactly. It creates a massive amplification loop that ultimately destroys the graft. But realizing that this exosome-mediated cross-dressing is what drives the T-cell reaction, that opened up a completely new therapeutic avenue.
Because if the foreign MHC on those circulating exosomes is the spark, we just need to take away the spark.
Precisely. The structural manipulation of those exosomes becomes the obvious intervention.
Which brings us to the core focus of today's deep dive. The profound, basically immunological silence of purified exosomes that specifically lack these MHC class 1 and class 2 proteins.
It's all about stealth mode. The sources detail some incredible in vivo models using exosomes derived from immature dendritic cells and also from human adipose mesenchymal stem cells.
I feel like the word immature is doing a lot of heavy lifting there. What's the difference?
It's critical. Mature dendritic cells are highly potent T-cell stimulators. Their surfaces are completely dense with MHC class 2 and necessary co-stimulatory molecules.
But the immature ones aren't.
No, exosomes from immature dendritic cells are 50 to 100 times less potent at stimulating a T-cell response.
Wait, 100 times just from being immature?
Yeah, and this massive drop in reactivity is entirely due to the absence of key surface proteins. These specialized exosomes lack MHC class 2, and they lack crucial co-stimulatory molecules, specifically things called B7.2 and ICM1.
Okay, so without the MHC class 2, there's no main target for the T cell to even recognize.
Right, no primary A antigen.
Mm-hmm.
And without B7.2 and ICAM1, the structural antennas required to physically dock with the T cell to deliver the secondary activation signal, they're just missing.
They just don't have the hardware.
Exactly. And the stem cell exosomes, the exo-AACs, have a similarly blank profile. No MHC class 2, no costimulatory molecules, and incredibly low or even completely absent MHC class 1.
So by using these inherently blank vesicles, the targets for Eller recognition are just gone.
Yeah.
The host's APCs have no foreign MHC to scavenge, meaning they don't cross-dress, meaning the T cells stay calm.
The entire threat is neutralized.
But wait, I have to ask a pushback question here because the immune system has backups, right? What about natural killer cells?
Yeah.
I thought they literally patrol the body looking for cells that are missing their native ID.
The missing self hypothesis.
Yeah, doesn't missing self trigger an attack anyway? Like, if a cell doesn't have MHC class I, the NK cells flag it as suspicious and destroy it. Shouldn't a blank exosome trigger that exact same response?
It's a really good question, and theoretically it should. But you have to remember, missing self is a threat response designed for whole living cells. Exosomes are nanoscale vesicles. They operate under completely different physical constraints.
Because they're just so small?
Exactly. For an MK cell to activate and release its destructive granules, it has to form a highly structured physical connection. It's called a cytolytic synapse.
It has to physically grab the target.
Right. But because these exosomes are merely 30 to 100 nanometers, and they lack the costimulatory array to stabilize a connection, they simply cannot form that cytolytic synapse.
The NK cell literally can't get a grip on it.
Exactly. The physical scale provides a built-in layer of evasion. It bypasses the tripwires that would normally shred a larger cell.
And since they lack MHC class 2 as well, they slip past the specific adaptive immune response, the helper T cells too.
They slip right through the cracks. But what's really amazing is that their function extends way beyond just passive evasion.
They aren't just hiding.
No, they are actively pacifying the host's immune system. The research details how these MHC-stripped exosomes exert a direct inhibitory effect on T-cell differentiation and activation.
Okay, but if they lack the surface proteins to dog normally, how are they communicating this?
It relies entirely on the transfer of their internal cargo. When these stealth exosomes fuse with the recipient's immune cells, They dump a dense payload of microRNAs and signaling proteins straight into the host cell's cytoplasm.
Oh, wow. So they just drop off the instructions internally.
Exactly. And that payload fundamentally reprograms the cell's behavior. The microRNAs specifically target and degrade the messenger RNA responsible for inflammatory cytokines.
So they turn off the alarm bells at the source.
Basically, yes. In vivo models show a drastic reduction in signals like interferon gamma and interleukin-17, which are the main chemicals that drive tissue damage in graft rejection.
They act like targeted gene silencers. They turn off the production before the cell can even make the inflammatory proteins.
Right, and simultaneously, they increase the production of anti-inflammatory signals, like interleukin-10.
That's incredible. So it's a complete shift in the chemical environment.
It is. And it even drives massive structural shifts in the immune cell populations. These exosomes actively promote the expansion of regulatory T cells, or TREGs.
And TREGs are the ones that calm down the other immune cells, right?
Yes. The exosomes dump regulatory microRNAs into standard P cells, forcing them to upregulate a transcription factor called FOXP3. This converts an aggressive effector T cell into a highly suppressive TREG.
Which creates like an immune privilege zone around the new organ.
Exactly. And they also target macrophages. During rejection, macrophages usually adopt an M1 phenotype, which is highly inflammatory and destructive.
They secrete those reactive oxygen species to just melt the tissue.
Right. But when these stem cell exosomes fuse with M1 macrophages, the internal microRNAs force a radical phenotypic shift into an M2 macrophage.
And M2 is the healing phase.
Yes. M2 macrophages are central to tissue repair. They promote collagen formation, blood vessel growth, and tissue remodeling.
So they're not just halting an attack, they are literally taking the destructive infantry and reprogramming them into a specialized repair crew.
It's a complete conversion.
So what does this all mean for you listening? It's like these exosomes aren't just stealth operatives sneaking past the security guards. They are stealth operatives who sneak in, hand the guards a cup of chamomile tea, and convince them to take the rest of the day off to do some gardening.
That is a very vivid, but honestly very accurate way to put it.
But wait, the bloodstream is still a really turbulent, hostile place. To do all this, they still have to survive the body's innate clearing mechanisms, right?
They do. They might lack the targets that trigger T cells, but they are armored with a highly specific array of defensive proteins. The sources highlight the dense presence of a protein called CD47 on these purified exosomes.
CD47, what does that do?
It's a critical regulatory protein. When a macrophage bumps into a circulating particle, its baseline instinct is to engulf and destroy it.
Phagocytosis. Just eat the trash.
Right. But when the macrophage's receptor binds to the CD47 protein on the exosome, it triggers a powerful inhibitory signal inside the macrophage itself.
It's like a biological kill switch.
Exactly. The CD47 interaction physically paralyzes the actin cytoskeleton of the macrophage. The complex structural machinery it uses to extend its membrane and swallow the particle is instantly locked down.
So the macrophage bumps into the exosome, but the CD47 physically prevents its cellular jaws from closing.
It just can't eat it. And they're similarly protected against the complement cascade.
That's the system that punches holes in foreign cells, right?
It's an ancient, rapidly acting network of plasma proteins. But these exosomes carry membrane-bound complement regulatory proteins, specifically CD55 and CD59.
How do those work against the hole punching?
Well, the complement cascade relies on a sequential amplification loop. CD55 disrupts the early stages by accelerating the decay of a key enzyme, preventing the cascade from gaining momentum.
It cuts the fuse.
Right. And if any part does manage to proceed, CD59 acts at the terminal stage. It physically binds to the assembling proteins and stops them from forming the final membrane attack complex.
So it literally stops the drill from breaking the exosome's lipid bilayer.
Exactly. So you have this combination. They lack allorecognition targets like MHC class I and II, but they retain these potent innate defense proteins like CD47, CD55, and CD59.
Which allows them to circulate with near impunity.
Yes. They survive the bloodstream long enough to locate host immune cells, fuse, and deliver the microRNAs that dismantle the rejection from the inside out.
Which is just a massive leap from the old way of doing things. I mean, traditionally, to prevent a host from destroying a transplanted organ, we just flood the patient with broad-spectrum immunosuppressants.
Indiscriminately shutting down the whole system.
Right. Which leaves them open to opportunistic infections, viruses, even malignancies. But utilizing these purified MHC blank exosomes presents a fundamentally different paradigm. We can induce this localized, highly targeted tolerance without crippling the patient's entire defense system.
By eliminating the proteins responsible for that semi-direct cross-dressing pathway, we prevent the spark.
Yeah.
And by delivering an active suppressive payload, we shift the local environment from hostile to completely accommodating.
So just to recap the aha moments for you guys listening, normally exosomes trigger transplant rejection by making our cells cross-dress in foreign MHC proteins. But by purifying exosomes that inherently lack MHC class 1 and class 2, like those from adipose stem cells or immature DCs, we create stealth vesicles. They bypass the NK cells because they're too small. They paralyze macrophages with CD47 kill switches. They block the complement cascade, and they actively rewrite the immune system to accept the new organ.
It's an elegant solution provided by the body's own biology.
It really is. And the long-term implications here, they have to extend way beyond just allotransplantation, right? Oh, absolutely. If we have isolated a naturally occurring, highly stable biological vesicle that can navigate the human bloodstream while remaining entirely invisible to the immune system, the therapeutic applications are virtually limitless.
Because you could theoretically load those empty stealth vesicles with almost anything.
Exactly.
Which leaves us with a pretty provocative thought to mull over. If we can master the use of these MHC blank stealth exosomes to stop the body from rejecting a foreign kidney or heart, could we engineer these exact same invisible delivery trucks to drop off targeted genetic therapies?
Like CRISPR for autoimmune diseases.
Right, bypassing the immune system entirely to correct a disease at the cellular level. Or even loading them with cancer-killing messenger RNA that can navigate straight to a solid tumor without being intercepted and destroyed by the body's clearance mechanisms.
Using a native immune-evasive vesicle solves one of the biggest pharmacological delivery challenges we face today.
It totally reframes how we interact with the human body. It's incredible stuff. Well, thank you so much for joining us on this deep dive. Thanks for having me. It was a great conversation.
And to everyone listening, keep questioning the microscopic world around you. Because, as the research clearly shows, the most powerful medical interventions are the ones the body never even sees coming.